Hello everyone, welcome back to the class. In our previous class, we started a new module that is cell cycle, and we discussed many aspects related to the main basic topics of cell cycle, right? To let us quickly revise what we learned in our previous class, that is the first class of the cell cycle. So cell cycle is an ordered series of events. It is a sequence of events by which a cell duplicates its genome and eventually divides into two daughter cells. It has two main phases, that is interphase and M phase. Interphase is known to be a resting phase, and it is a long phase, the time during which cell is preparing for division by undergoing both cell growth and DNA replication. M phase is a short period of 1 hour, and the period of M phase differs in organisms, and it is a period of actual division corresponding to visible mitosis. And also there is another cell division type will be discussed in the future classes, the phase which starts with nuclear division and corresponding to the formation of the daughter nuclei, that is karyokinases, and usually end with the division of cytoplasm, that is known as cytokinases. So this event is known to be cell cycle. Interphase is a resting phase. It is a very active phase where cell is preparing itself for the division by undergoing both cell growth and DNA replication in an orderly manner. So interphase is subdivided into three phases, that is G1 phase, S phase, G2 phase, and M phases, not the part of interphase. G1 phase is the gap phase between the period between end of the M phase and the start of the DNA replication phase. S phase is the period during the DNA where the DNA synthesizes, and G2 phase or gap 2 phase is a period following the DNA replication and preceding the initiation of M phase. G0 phase is another phase where the cells that do not divide enter into the G0 state. Cells are able to enter in three states: reversible G0 state, irreversible terminally differentiated state. Reversible G0 state is also known as quiescent state, where cells remain metabolically active but no longer proliferate unless the favorable environment is resumed. Irreversible G0 state is also known as senescent state, where the dysfunctional cells that have completely ceased proliferation and are permanently withdrawn from the cell cycle. It is said that it is irreversibly lost its proliferative potential. Terminally differentiated cell is the cell in this course of acquiring specialized functions have irreversibly lost its potential of division. So next, time duration, we also discussed 95% of the cell cycle is spent in interphase, and in G1 phase it takes 11 hours, in S phase it takes 8 hours, G2 phase it takes 4 hours, and M phase is the shortest phase, that is 1 hour. These time will vary across the organisms. So this much aspect we studied in our previous class. So today we'll be learning about the regulatory system, cell cycle control system. So cell doesn't divide itself automatically, so there is a particular controlling system, as children has their parents to control, right? So like that, the cell cycle is also having a control system that will be discussed today. Control system, what does it do? So it is an engine. Which helps in progression from one step to another step. Okay, it is a series of protein complex, we can say protein complex. So what is a controlling system in cell? So it is a protein complex. Protein complex is helping for the controlling the cell cycle. Okay, So the protein complex is made up of two subunits. They are cyclins and CDKs. Cyclin and CDK. I'll explore the CDK. CDK is nothing but cyclin dependent kinase. Cyclin dependent kinase. So there are two major subunits of the protein complex which are controlling the cell cycle. Okay, so they are what? Cyclins and cyclin dependent protein kinase. Remember this word kinase. Whenever you hear this kinase is an enzyme which catalyzes the phosphorylation. That means insertion of phosphate group to the substrate, to the component. So this kinase is an enzyme which catalyzes the phosphorylation reaction. Okay, so what are the two subunits of the protein complex we just mentioned now? That is cyclins and CDKs. Cyclin is a regulatory component. It regulates. It is a regulatory component. Then what about cyclin dependent protein kinase, that is CDKs? They are catalytic component. So cyclins regulate and CDKs catalyzes. As it is a protein kinase, it acts as a protein kinase. It adds phosphate to the proteins. Especially the proteins are made up of amino acids, right? So there is a particular amino acid. So this CDKs adds phosphate group to the particular amino acids. So cell cycle control system, it is an engine that drives the progression from one step to the cell cycle to the next are the series of protein complex. They are composed of two subunits, that is cyclin, which is a regulatory component, cyclin dependent protein kinase, that is CDK, which is a catalytic component. Here you can see a controller which is controlling. So this is a control system of a cell cycle. What and all it controls? It checks, is the DNA replicated in the G2 phase, is environmental favorable in a G2M transition. When it comes to M phase, are all chromosome attached to the spindle? It checks for that. And if we come to G1 phase, it still is the environment favorable to continue this cell division. So this is a control system. Here this image shows the two key components of the cell cycle control system. One is cyclin, which is mentioned in green color, and red color one is cyclin dependent kinase. So without this cyclin, CDK is inactive. So in the absence of cyclin, CDK is inactive. So in order to activate this cyclin dependent kinase, so CDK should be complexed with the cyclin. So where all you can see this system or this cell cycle control system? First we'll discuss in G1. Okay, G1, in this it checks for DNA damage. Is there any DNA damage? Okay, and here it checks for DNA damage or stalled replication, folks, same. And in G2 phase, it checks for damaged or unduplicated DNA. Okay, this is the time scale diagram which has already been discussed. Okay, so first we'll discuss cyclins in detail. Okay, we know that it is a regulatory component. regulatory component. Without this, the CDK remains inactive. So cyclin binds with the CDK molecule, and it controls phosphorylation action. Phosphorylation of a appropriate target protein. So each cyclin is rapidly synthesized. It is synthesized wherever needed, and also it is broken after its purpose. It just activates the cyclin CDK molecules or CDK molecules. After that, it breaks, and this is very similar to the signal peptides or the signal molecules which we discussed in the protein targeting class, right? So there is a signal peptidase where it cleaves the signal peptides or the sequence, targeting sequence or signaling sequence. So these signal peptidase will break that signal when the molecule has, or when the protein has been delivered to the particular destination, right? It is similar to that. So there are four classes of cyclins based on in which stage of the cell cycle they are present, at which stage they bind to CDK and functions. So first one is G1 cyclin. It is evidently showing this cyclin binds to the CDK at the G1 phase. Next one is G1S cyclins. This cyclin binds to the CDK at the period which is the transition period from G1 to S. Then is S cyclin, where this cyclin binds to the CDK at the S phase. Then M cyclin, where the cyclin binds to the CDK at the M phase or mitotic phase for now. Okay, this was about cyclin. Cyclin is a regulatory component which binds to the CDK, which rapidly synthesizes during the specific phase, and it is promptly broken down. There are four classes: G1 cyclins, G1 or S cyclins, and M cyclins. Now we'll discuss about CDKs. What is the CDK? It is cyclin dependent kinase. It is catalytic component, and it catalyzes which process? Phosphorylation process. Catalyzes the phosphorylation process. So to which amino acid of a specific protein does it adds a phosphate group? So first it needs to be activated. First cyclin binds to CDK, then it adds phosphate group to the protein. The protein is having serine or threonine. So to these they catalyze the CDK. So activated CDK catalyzes the phosphorylation of specific serine and threonine residue of the specific target protein. Okay, so target proteins of CDK phosphorylation are determined by the association cyclin based on the formation of cyclin and CDK complex at which stage. So they are classified into same as the cyclins, the four classes of CDK. CDK. When cyclin CDK complex disappear, the phosphorylated substrate proteins are rapidly dephosphorylated by enzyme phosphatases. So this is also very similar for reversible G0 state, which we discussed before. Okay, so as the CDK phosphorylates the protein in a particular residue, there is a reversible process where it dephosphorylates by enzyme phosphatases. Okay, so there are 4 classes of CDK. They are G1 CDK, happening in the complex of cyclin and CDK you come across in the G1 phase, and G1S CDK, it is found in the transition phase between G1 and S, and S CDK, which is found in synthesis phase, and last one is M CDK, which is these 4 classes are very similar to the cyclins. They are the 4 classes of cyclins. See, cyclins bind to CDK molecules and control their ability to phosphorylate appropriate target proteins. Each cyclins is rapidly synthesized during a specific phase of the cell cycle and is again promptly broken down after it serves its purpose. There are 4 classes based on in which stage they are present: G1 cyclins, G1 bar S cyclins, S cyclins, and M cyclins. Coming to cyclin-dependent protein kinase CDKs, so they constitute a protein kinase family. So kinase means it phosphorylates the proteins. These are enzymes that add phosphate group phosphorylation happening. CDKs are so named because their activities are regulated by cyclin. So it is cyclin dependent. Okay, they catalyze the phosphorylation of specific serine and threonine residues of specific target proteins. Target proteins for CDK phosphorylation are determined by the association cyclins. So different phases of the cell cycles are characterized by the phosphorylation of different target proteins. Phosphorylation events are transient and reversible. Once its job is done, it dephosphorylates. There are 4 different cyclin CDK complexes: G1 CDK, G1 bar S CDK, S CDK, and M CDK. So when cyclin binds to CDK, it forms a complex naturally, that is cyclin CDK complex, cyclin CDK complex, and it regulates the cell cycle, whether the cycle should stop or whether the cycle should run. That is controlled by this cyclin CDK complex. Okay, so there is another one separate regulatory protein complex that is known as APC bar C, which initiates metaphase to anaphase transition. So it is the complex or the controlling complex APC bar C which initiates the metaphase to anaphase transition, which will be learnt later. So this is also a control system. Okay. See, this image shows how the cell cycle control system varies the or oscillates during the cell cycle. So G1 bar S CDK is present at the G1 bar S cyclin level or in late G1 phase or in late G1 phase, and that controls what lead to the formation of G1 bar S CDK complex that trigger the progression through the start transition. Okay, then S CDK is initially present. It is present at the start of S phase and triggers the DNA replication, proper DNA replication. Then M CDK that is present in the G2 phase itself, but it is held inactive. Okay, they are activated during G2 bar M transition phase, and there is a separate APC bar C, as I said. So it initiates the metaphase and anaphase transition during M phase. So these are the cyclin CDK complex. Next, this table shows the major cyclins and CDKs of vertebrates and budding yeast. So cyclin CDK complex, that is G1 CDK, cyclin is cyclin D in vertebrates, the CDK partner is CDK4 and CDK6. Okay, there are 3 D cyclins in mammals, that is cyclin D1, D2 and D3. Whereas in budding yeast, the cyclin CDK complex G1 CDK, the cyclin is CLN3 and CDK1. In the G1 bar S CDK complex, the cyclin is E, CDK partner is CDK2. In budding yeast, the cyclin is CLN1, 2 and CDK partner is CDK1. So all CDK partner are CDK1 itself. Okay. In vertebrates, it differs. SCDK complex, the cyclin is A and CDK partner is 2 and 1. And in MCDK complex, cyclin is cyclin B and CDK partner is 1. So in our next class, we will be learning about the regulation activity of cyclin CDK complex. How the cyclin CDK complex regulates, or how it forms a destruction, or how it releases destruction and help in proceeding of cell cycle. That will be learnt in next class. Thank you.